Hello everyone, let's get started. Today we're going to be talking about the chemistry of biology. The way we're going to approach this lecture topic is by first talking about what we refer to as the biologically important elements and the molecules that are made from those elements. We're going to review the structure of the atom. We'll talk about different kinds of chemical bonds and attractions. We'll talk about some of the most important biological molecules, the molecules that are critical to cell structure and function. We'll talk about the concept of polarity, the importance of water, the different types of functional side groups that we find on many biological molecules. And we'll finish up by reviewing just a little bit about energy and the laws of thermodynamics. Many students find that this kind of chemistry of biology is difficult for them. The important thing for us to remember is that there are concepts in chemistry that are critical to our understanding of biology. We're primarily interested in chemistry where it overlaps with biology and helps us to understand cellular processes. We're interested in the key elements, the key molecules, the macromolecules, the bonds that form those molecules, and also how cells use energy. The good news for us as students of biology is that there are really 4 critically important elements inside of living cells. In fact, if you took all of the matter of all of the living things on our planet, 96% of it would be made out of only 4 elements: carbon, hydrogen, nitrogen, and oxygen. So while this periodic table is full of many different kinds of elements that would be very interesting to a chemist, we're more focused on these 4 critical elements. I've got the symbols here: carbon, hydrogen, nitrogen, and oxygen. Now, of course, there are lots of other elements that show up in living cells, other biological types of atoms. But remember, 96% of living matter contains just these 4. So it's important for us to understand how those 4 elements function, chemically speaking. On this slide, I've listed some of the key biological molecules, the molecules that are critical to the structure and function of living cells. Water is, of course, number 1, because cellular life, on our planet at least, is water-based. I've also listed the 4 macromolecules: carbohydrate, lipid, including fat, proteins, and nucleic acids. Underneath each one of these, I've written down a chemical structure. For example, under carbohydrate, I've written down C6H12O6. This is the chemical formula for the molecule called glucose. Glucose is a critically important carbohydrate inside living cells. The reason I wrote the formula out is because I want you to see what this molecule is made out of: carbon, hydrogen, and oxygen. 3 of the 4 key elements. Next on the list are the lipids. One kind of lipid is called fat. It's important to know that there are other lipids as well, so these 2 terms are not interchangeable. The example I've written down here is a molecule called cholesterol. Again, I've written out the formula so that we can see that it's made out of carbon, hydrogen, and oxygen, 3 of the 4 key elements. This ridiculous-looking chemical formula is underneath proteins. You would never write this molecule out this way, but I did it just for demonstration purposes. This is the chemical makeup of a molecule called hemoglobin. And over here on the right side of the slide is an image of what the hemoglobin molecule looks like as a three-dimensional folded protein. Look at what's inside hemoglobin: carbon, hydrogen, nitrogen, and oxygen, as well as 2 atoms of sulfur and 1 of iron. The vast majority of the atoms in this molecule are made out of the 4 key elements. Finally is listed nucleic acids. Remember, there are 2 kinds of nucleic acid in living cells, DNA and RNA. These nucleic acids have carbohydrate in them. I've written down deoxyribose and ribose. They have phosphate groups in them. They also have what we call the nitrogenous bases. These things are also primarily composed of the 4 key elements. Let's take a few minutes now and review what you've learned in chemistry about atomic structure. Remember that on the periodic table of the elements, each element is listed by name and by chemical symbol. This is the symbol that we use for carbon, uppercase letter C. Now, different versions of the periodic table present information differently, but on all versions, you're going to find 2 numbers associated with the chemical symbol for an element like carbon. You're going to find what's called the atomic number and what's called the mass number, or the atomic weight. Remember, every element has the same basic atomic structure. It has a tightly compressed atomic nucleus that's composed of positively charged protons, shown in red here, and neutral particles called neutrons. Protons and neutrons cluster together to create the atomic nucleus. Flying around that atomic nucleus are another kind of particle called electrons. Electrons are negatively charged. What the atomic number and the atomic weight, or mass number, tell us is just how many of these particles are present in this particular element. The atomic number tells us the number of protons. There are 6 in carbon. And the mass number, or atomic weight, tells us the combined number of protons and neutrons, and there are 12. So that means we have 6 protons and 6 neutrons, because 12 minus 6 is 6, in the atomic nucleus of carbon. The other thing we can tell from this information is how many electrons there are. Because in a neutral atom of carbon, in other words, no charge, no positive or negative charge, we know that the number of electrons is equal to the number of protons. If there are 6 protons, there are 6 electrons. The other thing we need to remember about the elements is that sometimes there are versions of each element. For example, most of the carbon on our planet is referred to as carbon-12. The mass number is 12, again, 6 protons and 6 neutrons. But there's some carbon on our planet that's called carbon-14. This version of carbon has 2 extra neutrons in its atomic nucleus. When we have 2 different versions of an element, we refer to them as isotopes. When it comes to atomic structure, in biochemistry, we spend a lot of time talking about electrons. Remember, electrons are in motion around the atomic nucleus. It might appear that the electrons are sort of flying randomly in a cloud, but in reality, we can make predictions about where these electrons will be at any one moment in time. Electrons tend to move around within discrete regions around the atomic nucleus that are called orbitals. Now, there are lots of different orbitals. We don't need to understand much more about orbitals other than they are regions around the atomic nucleus that hold electrons. And each orbital can hold up to 2. Orbitals that surround the atomic nucleus are combined together into levels that we call electron shells. Each electron shell then contains some number of orbitals, and each orbital contains up to 2 electrons. So if we know how many orbitals are in a shell, we know how many electrons can fit in that shell. We simply multiply the number of orbitals by the number 2. 2 electrons per orbital. If a shell has 4 orbitals in it, we multiply 4 by 2, and we know that that shell can hold 8 electrons. On this slide, you're looking at a drawing of all the different orbitals that exist around an atomic nucleus. Again, we're not really interested in learning all of the different orbitals and all of their symbols and shapes. What we need to understand is that these orbitals get grouped together into what we call electron shells. The reason we care about shells is because the outermost shell has a lot of importance in biology. The outermost electron shell is referred to as the valence shell. If you look at this drawing over here on the right, this represents a copper atom. In the center of the drawing is the atomic nucleus. Remember, here we're going to find the protons and the neutrons tightly clustered together. Outside of the atomic nucleus are the electrons in motion in a cloud. At any one moment in time, we can predict where those electrons are going to be in terms of orbitals that surround the atomic nucleus. We group the orbitals together into shells, and we draw these shells as concentric rings. So here's one shell. The 2 black dots represent the pair of electrons that can be found reliably in that shell. Here's the second shell. It has more electrons in it because it has more orbitals in it. Here's the third shell, even more electrons. The final shell, the outer shell, and it's going to vary in number depending on the element. This outermost shell is called the valence shell. The reason that we care about this valence shell is because sometimes the valence shell will contain an unpaired electron. Unpaired electrons are unstable. In other words, any atom that has an unpaired electron in its valence shell is chemically unstable. And what we mean by that is that it is available to participate in a chemical reaction. You're going to hear me say time and again in this course that nature seeks stability. Nature would prefer that every atom be stable and have paired electrons in its valence shell. Atoms are most stable when their valence shell, in fact, is full, when all of the orbitals in the valence shell are full of electrons. Now, the way that valence shells can become full is when atoms interact with each other. That's how covalent bonds are formed between atoms. So, for example, if 2 atoms each have an unpaired electron in their valence shell, both of those atoms are unstable. The way that they can become stable is by joining together and sharing those unpaired electrons. Each atom has 1 unpaired electron, so if they share them, now they each have a pair of electrons in their valence shell. That's all a covalent bond is. A covalent bond is defined as a pair of shared electrons between two atoms. Remember from chemistry that covalent bonds are very strong, they're very stable, because those two atoms are more stable when they're bonded together like that than when they're separate. It's covalent bonds that create molecules inside of cells. In fact, it's covalent bonds that create all molecules. Molecules like hydrogen gas or water. Now, up until this point, most students can conceptualize the idea that two atoms can come together and share the unpaired electrons in their valence shells. Where it gets a little bit complicated is this next bit of information, and that's that sometimes when two atoms share a pair of electrons, they don't share those electrons equally. Atoms have a property, a chemical property, referred to as electronegativity. Each element has its own level of electronegativity. What electronegativity describes is the ability of a particular atom of an element to pull on a pair of electrons and bring them closer to the atomic nucleus. Elements with a higher electronegativity have a greater pull. They're able to bring that shared pair of electrons closer to them and farther away from the other atom. Now, if we look at our four key elements, we can put them in order based upon their level of electronegativity. And that's what I've written here. Oxygen has the highest electronegativity of the four key elements. It's higher than nitrogen and carbon and hydrogen. Notice that I've got an approximately equal sign between the carbon and hydrogen here. Technically speaking, there's a slight difference in electronegativity between carbon and hydrogen, but it's so slight that we don't consider it to be important in biology. We consider carbon and hydrogen to have the same electronegativity. So, first comes oxygen. It has the highest electronegativity. Then comes nitrogen. And then comes both carbon and hydrogen. They have the same low level of electronegativity. When you bring two atoms together that have a different level of electronegativity, that's when you start to see this unequal sharing of electron pairs. Anytime there is unequal sharing going on, we give that covalent bond a name. We call it a polar covalent bond. So a polar covalent bond is simply a pair of electrons that's being shared between two atoms, and those electrons are being shared unequally. One atom is holding the electron pair closer to it and farther away from the other atom. Sometimes two atoms come together and they share a pair of electrons perfectly equally in a covalent bond. We also give that type of bond a name. That's referred to as a nonpolar covalent bond. On this slide up at the top, you're looking at a drawing of hydrogen gas, H2. And the artist has drawn the two individual atoms of hydrogen with a covalent bond between them. Remember, in chemistry, when we draw a molecule this way, we draw the covalent bond as a solid line. Now, in this drawing, we've added in the electron pair to remind us that that's what this line is. This line represents a pair of electrons that's being shared between these two hydrogen atoms. Now, I said a moment ago that hydrogen has a low level of electronegativity. If we put two hydrogen atoms together, they're each going to pull on this pair of electrons. But they're going to pull in equal and opposite directions because they're the same type of atom. That means that that electron pair is going to sit right in the center of that covalent bond. And that's what we've drawn here. In other words, this pair of electrons is being shared equally between these two atoms, and we would call this a nonpolar covalent bond. Down at the bottom, we have a similar drawing, but this time it's a water molecule. There are two covalent bonds in this molecule, and we have an oxygen to hydrogen bond here and an oxygen to hydrogen bond here. This drawing looks different though, because the two pair of electrons are not sitting in the middle of each of those covalent bonds. Instead, they're drawn sitting very close to the oxygen atom. Remember, oxygen has a much higher electronegativity than hydrogen does. So oxygen is going to pull on the electron pair in each covalent bond more strongly than the hydrogen will. And those electrons are essentially going to sit much closer to the oxygen than to the hydrogen. These are what we would call unequally shared electrons, and therefore these are polar covalent bonds. Now there's something else in this drawing that's important to us, and that's these little symbols. This symbol is a lowercase d in the Greek alphabet, so this is delta, and you can see it's delta minus here and delta positive next to the hydrogens. These symbols tell us that this molecule contains what are called partial charges. The oxygen is partially negative. The hydrogens are partially positive. Now it's important to remember that a partial charge is not a full charge. Water is a neutral molecule. It does not have any charge associated with it. But inside a water molecule, there's an area that's relatively more negative, and there's areas that are relatively more positive. And it's all because of where the electrons are sitting. Remember, electrons are negatively charged. So if you're an atom and pairs of electrons are sitting very close to you with their negative charge, you have become relatively more negative. In comparison, atoms that are farther away from those electrons are now relatively more positive. Polar covalent bonds create partial charges inside of molecules. On this slide, I've just written out in words what we just looked at in the diagrams. Polar bonds, polar covalent bonds, produce partial charges. For example, in a water molecule, oxygen has the higher electronegativity compared to hydrogen, so the oxygen atom is going to pull the electron pair closer to it and farther away from the hydrogen. Oxygen becomes partially negative, hydrogen becomes partially positive. Therefore, in a water molecule, we have two polar covalent bonds. Water is therefore a polar molecule because it contains partial charges. If we compare that to a molecule like methane, we see something very different. Carbon and hydrogen have approximately the same electronegativity. So when we create 4 covalent bonds between carbon and hydrogen, and that's what we have in a molecule like methane, we have 4 covalent bonds. the electron pairs in each one of those bonds is going to be shared equally between the carbon and the hydrogen. Therefore, we have nonpolar covalent bonds in that molecule. No partial charges are forming, so we have a nonpolar molecule. Now, why do we care about this? We care about this because polar molecules are going to behave very differently than nonpolar molecules inside of a living cell. Polar molecules have a different chemistry, in other words, than nonpolar molecules do, and so they're going to behave differently. And that's going to be important with regards to both structures inside of a cell and the way the cell functions. Now, in addition to covalent bonds, there are several other types of chemical bonds that we need to be familiar with in biochemistry. The first of these is referred to as ionic bonds. Ionic bonds form between ions. And all an ion is an atom that has either lost or gained an electron. So when it comes to ionic bonding, we're no longer talking about two atoms that are sharing a pair of electrons. Now we're talking about a pair of atoms who are literally transferring electrons one to the other. One of those atoms is going to physically lose the unpaired electron that's inside its valence shell. And the other atom is going to gain that electron. A good example of this is the molecule called sodium chloride, or table salt. Sodium chloride contains an atom of sodium and an atom of chloride. Those two atoms are not covalently bound together. They're bound together with an ionic bond. Both of these atoms have an unpaired electron in their valence shell. But instead of coming together and sharing that pair, the sodium is going to literally give up the unpaired electron in its valence shell and transfer it to chloride. Now, the result of this is that the sodium is now going to have one fewer electrons floating around its atomic nucleus. Remember, in an uncharged atom, the number of protons and the number of electrons is going to be equal. If an atom loses an electron by giving it away, it now has one more proton than electron, and it takes on a positive charge. What's happening to chloride is different. Chloride also has an unpaired electron in its valence shell. Instead of sharing that electron with another molecule, chloride is going to receive an extra electron from sodium. That means that the chloride atom now has one extra electron. So there's one more electron compared to the number of protons, and chloride takes on a negative charge. When an ion forms from an atom losing an electron, that ion has a positive charge, and we call that ion a cation. When an ion forms from an atom gaining an extra electron and gets a negative charge, we're going to refer to that as an anion. And when two ions form a bond like this, that bond is referred to as an ionic bond. Now, ionic bonds are very important, but they're not as strong as covalent bonds. If you take a molecule of sodium chloride and you put it into water, the sodium chloride is going to dissolve. The ionic bond is going to be broken, and the two ions will separate from each other. The reason that happens is because of the partial charges that are inside of water molecules. Remember, the oxygen in a water molecule is partially negative, and the hydrogens are partially positive. So the partially negative oxygen atoms are going to pull on this sodium. They're going to be attracted to it because the sodium has a positive charge. The negative oxygen is going to be attracted to the positive sodium, and it's going to pull on it. In comparison, the partially positive hydrogens in the water molecule are going to be attracted to this negatively charged chloride, and they're going to pull on it. And the result is that the sodium chloride is going to separate, it's going to break apart. The ionic bond will be broken. On this slide, you're looking at a visual depiction of this transfer of electrons between ions in an ionic bond. So up at the top, we're looking at a sodium ion being formed, and down at the bottom, we're looking at a chloride ion being formed. If you look, the sodium has an atomic nucleus, just like all atoms do, and then it has these concentric rings around it. These are the electron shells. Notice that there's a pair of electrons in the first shell. There's 1, 2, 3, 4 pair in the second shell. It's the outer shell that we're interested in. That's the valence shell. And in the valence shell, there's an unpaired electron. Down at the bottom, if we look at the chloride atom, we see something similar. Now, the number of electrons is different in a chloride atom than in a sodium atom because they're two different elements. But if we go to the valence shell, we can see that there's a pair of electrons here, there's a pair here, there's a pair here, but here's an unpaired electron. When the chloride atom receives the electron being donated by the sodium atom, the problem is solved. Now it has a pair of electrons in that valence shell. Now it has become more stable. But it also has an extra electron that it didn't used to have, so it takes on the negative charge. When a bond forms between two ions, it's called an ionic bond. It holds them together, but not nearly as strongly as a covalent bond does. On this slide, you're looking at a diagram of what's referred to as the electron sharing continuum. It depicts for us the different ways that two atoms can come together and deal with the problem of having unpaired electrons in their valence shell. If we start over here on the left, we have sort of one extreme, and that is that those two atoms could come together and they could share that pair of electrons equally between them. When two atoms have the same electronegativity and they share the unpaired electrons in their valence shells, that pair of electrons will be shared equally or symmetrically between them. That's what we call a nonpolar covalent bond. You can see it in this hydrogen gas molecule, and you can see it in this methane molecule. Carbon and hydrogen have equal electronegativity, so when they share a pair of electrons, they're going to share them equally. Next over, as we travel down this continuum, is the example of the polar covalent bond. The two atoms are still going to share their unpaired electrons in their valence shells, but they're going to share them unequally. This is a representation of ammonia, and here's our old friend water. Nitrogen has a higher electronegativity than hydrogen does, so the nitrogen is going to pull each one of these electron pairs closer to it and farther away from the hydrogen atom. Because electrons are negatively charged, that's going to make this nitrogen atom relatively more negative. And the hydrogens that are now further away from the electron pairs are now partially positive. Polar covalent bonds create partial charges. Unequal sharing of electrons, or asymmetrical sharing of electrons, creates partial charges, and molecules that contain partial charges are polar. On the other extreme of this continuum, we have the transfer of electrons. Two atoms are going to come together, and they're not going to share. They're not going to share equally. They're not going to share unequally. They're going to actually transfer the electrons. The sodium atom, which has an unpaired electron in its valence shell, is going to donate that electron to the chloride. Chloride also has an unpaired electron in its valence shell, so when it receives the electron from sodium, it now has a full pair. Because sodium loses an electron, it becomes positively charged. Because chloride gains an electron, it becomes negatively charged. The bond that forms between two ions is an ionic bond. There are two other types of bonds that we need to be familiar with. One is called a hydrogen bond, and the other is called van der Waals interactions. These types of bonds are weak bonds, also called attractions, that occur between two different molecules or even two parts of the same molecule. Hydrogen bonds are built when two polar molecules come together, or two polar parts of the same molecule. It's an attraction that is built between partial charges. Partially positive atoms or regions within a molecule are attracted to partially negative atoms or regions of a molecule. What results is a weak bond or a weak attraction called a hydrogen bond. The reason hydrogen bonds got their name is because one of the atoms is almost always a hydrogen. We can see hydrogen bonding in action in the DNA molecule, because the two strands of DNA that come together to form the double helix are held together through hydrogen bonding between the base pairs. Now, van der Waals interactions are different. They're also weak interactions, but this time they occur between two molecules or two parts of the same molecule that are nonpolar. A good place to see this is in protein folding. Remember, proteins are amphipathic. Proteins contain both polar and nonpolar regions. So the nonpolar regions within a protein molecule will be attracted to each other and held together through weak interactions called van der Waals interactions. We can use the water molecule again as an example of hydrogen bonding. When water molecules come together, because they're polar, they contain partial charges. And what will happen is the partially negative regions, the oxygens, will be attracted to the partially positive regions, the hydrogens, and we'll get a hydrogen bond formed. Notice that we draw hydrogen bonds as dotted lines, as opposed to the solid lines that we draw as covalent. The dotted line reminds us that a hydrogen bond is not as strong as a covalent bond. On this slide, you're looking at a depiction of protein and how proteins fold up. The folding that happens in a polypeptide in order to make a protein, the folding is the result of these types of weak interactions. If you look at the top of the drawing, this long green ribbon represents a polypeptide. So remember, a polypeptide is simply a long chain of amino acids that are bound together. The bond that joins amino acids in a polypeptide is a covalent bond, and it gets a special name. We call it a peptide bond. So this chain, this polypeptide, this is a very strong strand of amino acids. But remember, polypeptides don't stay as chains in living cells. Polypeptides fold up to create proteins. And the folding is the result of weak interactions. What they're showing us here is hydrogen bonding going on. There's an amino acid over here that has an R group, a functional group, that has a hydrogen in it. Over here in another spot on the chain, there's an amino acid that has a nitrogen in it. The nitrogen, which is going to be partially negative, and the hydrogen, which is going to be partially positive, are attracted to each other, and they'll be hydrogen bonded together. In other parts of this polypeptide, there are amino acids that have nonpolar functional groups, nonpolar R groups. We can look at these side groups and know that they're nonpolar because all they contain is carbon and hydrogen. Carbon and hydrogen have the same electronegativity. So this region of the amino acid is nonpolar, and so is this one. Those nonpolar regions are going to be attracted to each other and held together with van der Waals interactions. There are other areas of the polypeptide that are going to be drawn together through ionic bonding because regions of the molecule, the functional groups on different amino acids, are sometimes charged. The positive charge will be drawn to the negative charge and an ionic bond will form. There's even an example of something called a disulfide bond here between two sulfur atoms. We don't talk about the disulfide bond in this course. It's not nearly as common as hydrogen bonds and van der Waals interactions. Down at the bottom of this slide, you're looking at drawings of different proteins. If you Google any protein and you look for its structure, you'll often see a drawing that looks like this. These drawings are showing us the three-dimensional shape of the protein, drawn to indicate where different specific, what we call secondary structures are forming. If we go back to the green ribbon, sometimes hydrogen bonding creates what we call an alpha helix. If enough hydrogen bonding goes on in one particular area of the polypeptide, you get this helical structure forming. There's another specific kind of fold that occurs in protein called a beta sheet or a beta pleated sheet. Instead of finding a twisting helix, we find sort of a gently turning, two-dimensional, flat sheet. That's being indicated here by these arrows. This is a region of the protein where we also have interactions going on, but these interactions keep this area flattened as opposed to twisted into a helix. Proteins form through combinations of alpha helices and beta sheets. Now, it's important for us to step back here and remind ourselves why we care about all this. Why do we bother learning this biochemistry? Why do we bother learning or relearning the kinds of chemistry that relate to biology? We care about it because it turns out that living cells and the organisms that are made from those cells actually contain relatively few different elements within them. In fact, there are really 4 key elements in living cells: carbon, hydrogen, oxygen, and nitrogen. 96% of all of the mass of living things is made out of those 4 elements. All of the molecules that are built from atoms of those elements make up all of the different molecules inside of cells. You can take those 4 elements, in fact, and you can create quite an array of very complex, very important molecules, the kinds of molecules that help keep cells alive. All of that complicated structure supports all of the complicated physiology inside of living things. Those 4 key elements allow for all of the different metabolic reactions, respiration, photosynthesis. They allow for things like physical movement, sensation, nerve transmission, cell division, you name it. We care about biochemistry because it helps us to understand the workings of living things. We're going to change gears here a little bit and take a closer look at this molecule called water. I said earlier that living cells are based upon water. All life on our planet is based upon this important molecule. It is essential for life on our planet. We know that through evolution, the very first life forms developed in the marine environment. Eventually, those living things evolved to a point where they could leave the oceans and come up onto land. But we all still carry the memory of our evolutionary past because all of our cells contain a lot of water. Not just plain water, though, salt water. The salty water that makes us up reminds us that our evolutionary ancestors came out of the seas. Humans are, in fact, made up mostly of water, anywhere from about 65 to 75% water, again, salt water. Depending on the stage of life that the human is at, the amount of water varies a little bit, but we are primarily composed of water. We know that water is a highly polar molecule. It contains partial charges. We know that polar things can interact with and bind to other polar things because of those partial charges. When those polar things come together, they form hydrogen bonds. Water will readily hydrogen bond with other polar molecules or parts of molecules. That makes water an excellent solvent. Water is capable of dissolving other polar substances. In fact, we refer to those other polar substances as being hydrophilic or water-loving. Polar things like to be with other polar things. And since water is polar, other polar things like to be with it. They are hydrophilic, water-loving. This also explains the expression like dissolves like. A polar solvent will dissolve polar molecules. We also know that things that are non-polar do not interact well with water because, again, water is polar. So non-polar molecules will be repelled by water. They'll be isolated by water. They will not interact with it. So we call them hydrophobic or water-fearing. On this diagram, we can see a drawing of a salt molecule, sodium chloride. And then we see that molecule being placed into a watery environment. like inside of a cell. Remember, water is polar, and so it contains partial charges. And those partial charges are going to be attracted to any kind of molecule that ionizes, like sodium chloride does. Remember, what holds sodium chloride together is ionic bonds. And when you put sodium chloride into water, those ionic bonds can be broken. The relatively negative oxygen atoms in the water molecules are attracted to the positively charged sodium, and they will pull on it. The partially positive hydrogen atoms in the water molecules are going to be attracted to the negatively charged chloride atoms, and they're going to pull on those. That's going to help break the ionic bond that holds sodium chloride together. That's going to help sodium chloride dissolve in water. In comparison, if we took nonpolar molecules, like the octane molecules drawn here, and we put them into water, something very different happens. These molecules are called hydrocarbons. They're made out of carbon and hydrogen, the little white circles. That's all that's in them. Carbons, the black circles, and hydrogens, the white circles. And those are covalently bonded together. These are nonpolar molecules. Carbon and hydrogen have the same electronegativity, and so they're going to pull on the electron pairs equally in opposite directions. And we call that equal sharing nonpolar covalent bonds. There are no partial charges in nonpolar molecules. So when they interact with water, there's no way for them to bind together. Water contains partial charges. Water will be attracted to other polar molecules, like other water molecules, and will hydrogen bond. The partially negative oxygens will hydrogen bond with the partially positive hydrogen atoms. They don't know what to do with these nonpolar molecules. There's no way for them to interact. So the nonpolar molecules get isolated. They will come together, they will sort of hold together, but they cannot interact with the water around them. They cannot dissolve. So when we use this term polarity in biology, we're often talking about this characteristic, this asymmetry that we see in some molecules. Molecules that contain polar covalent bonds also contain partial charges. They are asymmetrical because of those partial charges. We often describe polarity also as being directionality. All that word means is that inside of a molecule, different regions behave differently. They look different, they behave different because of those partial charges. It turns out that when we look at our 4 macromolecules, several of them are polar, at least overall. Proteins, for example, tend to be polar overall. In other words, they tend to have more polar regions than nonpolar regions. Some proteins, though, are truly amphipathic. Remember, when we use that word, we're saying that the molecule has both polar and nonpolar chemistry. Carbohydrates also tend to be polar overall, as do the nucleic acids. The important exception among the macromolecules, therefore, are the lipids. Lipids tend to be nonpolar overall. Lipids are hydrophobic, water-fearing. Remember, though, there's a particular kind of lipid called a phospholipid that is amphipathic. It really does have both polar and nonpolar chemistry going on at the same time. It's the phospholipids, remember, that make up our cell membranes. Let's switch gears now and talk a little bit about what we sometimes refer to as biological molecules and their chemistry. All of the living things on our planet are said to be organic. Remember, in science, the word organic means carbon containing. So all of the molecules that make up all of the cells inside all the different organisms that live on the planet Earth are organic. They are built upon this important element called carbon. Carbon, therefore, is essential for life and is found in all 4 of the macromolecules. We often refer to carbon as being the skeleton of the macromolecules. When you look at any biological molecule, the things that are attached to that carbon skeleton can sometimes be referred to as side groups. Side groups or functional groups or R groups are all the same thing. Those terms all mean the same thing. Take a look at this molecule at the bottom of the slide. This is a drawing of an amino acid. All amino acids have the same basic structure. Remember, there are 20 different amino acids that make up all of the different kinds of proteins inside your body. All amino acids have a central carbon, and that carbon can create 4 covalent bonds. One of the bonds will be to a singlet hydrogen. One will be to a functional group called an amino group. One will be to a functional group called a carboxyl group. And the last one is labeled R because the last one is going to vary depending on the amino acid. So all amino acids have the central carbon, the hydrogen, the amino group, the carboxyl group, but the R group is going to be different from one amino acid to another. It turns out that there are 6 of these functional groups, these side groups or R groups, that are really important to us in biology. So important, in fact, that we need to know the name of these 6 groups and what the structure looks like. Now, on this slide and the next, we're going to see those 6 groups. These tables, though, contain some information that we don't need to know right now. So we need to know the name of the group. That's shown over here in the left-hand column. We need to know what it looks like structurally. That's shown in blue. And then we can ignore the rest of the information. The first functional group to know is the amino group again. An amino group is composed of a nitrogen and 2 hydrogens. Next on the list is the carbonyl group. A carbonyl group can actually take 2 different forms. It depends whether or not it's occurring at the end of a molecule or somewhere in the middle. So a carbonyl group can either be a carbon, an oxygen, and a hydrogen, or a carbon and an oxygen. The carboxyl group looks similar to the carbonyl, but a little bit different. It's a carbon, 2 oxygens, and a hydrogen. Next is the hydroxyl group. That's an O and an H, oxygen and hydrogen. Next is a phosphate group. This one is particularly important to us. We'll talk about this a lot when we talk about energy. A phosphate group is composed of a phosphorus atom and 4 oxygens. And finally, we have the sulfhydryl group, which is a sulfur and a hydrogen. We don't talk about sulfhydryl groups as much as the others. And that brings us to the last thing that we'll talk about in this lecture, which is just a little bit about energy. We're going to be talking about energy in more detail in a couple of weeks, but today we'll talk about energy as it relates to this biochemistry topic. The first thing we need to understand is what this word means. We tend to use the word energy in our casual language every day. People generally talk about having energy or not having energy, feeling very tired or feeling very energetic. But we need to have a definition for this word. And we use the formal definition, which is the ability to do work. Now, when it comes to cells, we can see the ability to do work in different forms. We can see what we refer to as movement energy or kinetic energy. We shorthand that as KE. We can see heat energy or thermal energy. We can see energy that is being stored inside a covalent bond. That's what we call potential energy. The important thing to remember is that in a living cell, it's the electrons that are found around atoms and in molecules that are the conduits of energy, the carriers of energy, if you will. When a pair of electrons is sitting in a covalent bond, the energy that's associated with them is being stored there. It's not available right now to the cell to do work. In order to obtain the energy in those electrons, the bond would have to be broken. Cells, it turns out, are very good at transforming energy from one form to another. Mechanical energy, thermal, sound, electrical, whatever it is, cells are very good at transforming it from one to another. Now, it can be a little bit confusing for us as students of biology when we read different source materials concerning this topic of energy. And that's because the terminology around energy tends to overlap. For example, when we talk about stored energy in a covalent bond, some sources will refer to it as chemical energy. Others will refer to it as potential energy. Both of those things mean exactly the same thing. Chemical energy, or the energy that's found in a covalent bond, is also being stored. It's also potential energy. As long as we remember that there's some overlap in the language, we can remember the more important things, the bigger picture things. That's what's written down at the bottom of the slide. Remember, all of the chemical reactions that go on in a cell, altogether they're referred to as metabolism. And we can group those chemical reactions according to whether or not bonds are being built or broken apart. Anabolic reactions build covalent bonds. Catabolic reactions break them. When bonds are broken, the electrons in those bonds are set free and energy is released. When bonds are built or formed, the electrons are being stored in the bond. That may require energy in order for its formation. Breaking bonds releases energy. Building bonds requires energy. Catabolic versus anabolic metabolism. Again, we're going to talk about energy in much more detail as we move forward in the course. The last thing I want to talk about before we finish up our lecture topic today is a little bit more about thermodynamics, the laws of thermodynamics. Remember the first law, the law of conservation of energy, tells us that energy can neither be created nor destroyed. You cannot create it out of thin air, and you cannot make it go away. All a cell can do is alter it in its form. And this is what cells are very good at. They can't create it, they can't destroy it, but they can transform it. They can alter it in its form. Now, what does that mean to us? What does that really mean? In a biological way, from a biological perspective, it tells us that all of the energy that is within a cell has to either be stored in the covalent bonds or released into the surroundings. That would be actually lost to the cell as heat. And finally, it could be transformed, moved from one type to another type. Cells have those three options, and that's it. They either store it, they transform it, or they allow it to be lost to the surroundings, to the universe as heat. You've heard me say that cells are very thrifty. Cells don't want to waste energy. So they put a lot of time and effort into harvesting energy and storing it until they need it, so that energy will not be lost to the universe as heat. The last thing we'll touch on today is the second law of thermodynamics. This is the law of thermodynamic spontaneity, and this is the one that students tend to not really like. It's a little harder to conceptualize. The second law of thermodynamics tells us that the universe is always tending toward greater disorder, what we think of as randomness or chaos. And the technical term for this is entropy. Now, what does this law tell us about biology? It tells us that the universe is tending towards fewer covalent bonds, and therefore smaller molecules. Why does that matter? It matters because living cells are working against this. It takes work, it takes energy to build more and more bonds and larger molecules, the macromolecules that cells need in order to survive. The universe is tending towards fewer covalent bonds, smaller molecules. Cells are working against that, and that requires energy.